Great, thank you so much. Thank you everyone for coming to my talk. Thank you to the organizers. This has been a wonderful meeting. This is my first time in this meeting. So yeah, I really appreciate everything I have learned here. And I know what the type of work I do is slightly different from what you do. So let's see how this works. Okay, so well, my title, the idea is to use machine learning methods or statistical methods to try to understand genome-wide association study loci. So how many of you know what a GWAS is? Most people, yeah, that's good to know. But I'll give a brief explanation the way I look at it. But anyways, so well, genome-wide association studies have been being performed by the field for the last more than 15 years. We keep discovering more and more loci associated with more and more traits. As of April this year, we had hundreds of thousands of loci associated with different traits at genome-wide significance level. But as you probably all know, most of the variants are located in non-coding regions, so we don't really know for the majority of this which one is even the causal gene, right? So back in the days, like 10 years ago, in order to try to address this problem, I proposed a method that I called PredictScan, which is now called transcriptome-wide association studies. So as you can see, I'm calling it PredictScan 1.0, so you see where I am going with this, right? So, but I will give you an introduction. So how many of you know what a TWAS is, or transcriptome-wide association study? So yeah, a good amount of people, but okay. So what is a GWAS? So I think of a GWAS, you have this matrix of, let me see if I can use this, of genotype matrix. Rows are people, columns are genetic variants. I just call them SNPs to keep it simple. And then you have your vector Y of phenotypes, so with the same number of rows as the genotype data. And a GWAS is the simplest thing you could ever think of doing with this data, which is a linear regression of your phenotype Y on each one of these columns, one at a time. Maybe, in a pinch, you do logistic regression. So you end up with this SNP level table of results where your column is, I like to call the effect of the SNP on the phenotype delta, because that way it doesn't get confused with the effect of the SNP on the gene expression levels. So, okay, so this is the effect size of the SNP on the phenotype. I have to find the right distance, sorry. You have the standard error column and the p-value. Small p-values are good because you get to publish. So that's good. But as you know, SNPs, you get RS 1, 2, 3, 4. You don't know what they do for the most part. So why not run a gene level association study? So you can measure transcriptome and run association between your phenotype and gene expression levels, one gene at a time, the same way you do it with the genotype matrix, and you would end up with gene level association table. So that would be more informative than the SNP level association table. But so what are the problems with that? So RNA sequencing is very expensive, right? It's like genotyping is cheap, RNA sequencing is a lot more expensive. There are many tissues like brain, pancreas that are very hard to access, so you couldn't get enough sample sizes. And more important, or as important, is that the differentially expressed genes that you find may be a consequence of the disease and not the cause of the disease. So this reverse causality makes this method kind of less optimal, right? So in order to sort of address that problem, we proposed over 10 years ago to take the genotype data and predict the expression levels. So instead of using observed transcriptome, we're going to use genetically predicted transcriptome. And then do the same thing. You take your Y, you regress on each one of those columns, you end up with gene level association study. So that's TWAS, transcriptome-wide association studies. We called it at the time PredictScan. It's one of the many methods that have been developed since it was first proposed. Okay, so how you train these prediction models, right? So we use reference transcriptome data, for example, GTEx, where you have genotype data, so the same type of matrix typically smaller sample size, right? They are not as big as the GWAS sample sizes. And then you have transcriptome loads, so RNA sequencing data for, in GTEx, you have over 50 different tissues. So rows are people again, columns are different genes. So each column is going to be the gene expression level for a given gene at a given tissue. So all you do is you train this linear model, kind of regularized regression model where T is the expression of the gene, so one column for a given tissue. You can make it more sophisticated, but in the end, it doesn't make that much of a difference. And you come up with this prediction model that is just linear combination of your SNPs. These W's give you the ways that we save in a database that we are calling PredictDB, and you can access it and download prediction models for all sorts of different gene expression levels in different tissues and so on. So how well do we predict this gene expression? And I am giving you here 4 examples, cherry-picked examples where we do particularly well. So on the x-axis, you see predicted expression levels. On the y-axis, you see observed expression levels. Here, the dots are people, right? That's going to be important to make a difference later. So for these 4 genes, you can see that we do a pretty good job at predicting gene expression levels with R-squares pretty high. So, but across the genome, if we look at all the genes, here I ordered the genes by their heritability estimates, so heritability of the expression level of the gene, and you can see, and in red dots are how well we are predicting the observed values, right? So the more heritable the gene expression is, the better we are predicting, which makes sense. So this is just reassuring that we are not, making things up, and this is just very consistent. So there are many advantages in running a gene level association. I'll go quickly through it so we can get to the other aspect later. So for example, reduce multiple testing burden. So instead of having 1 million or 10 million SNPs that you have to correct for, you only have, let's say, 10,000 genes, because we don't really, with these population-based methods, we don't really predict many more than 10,000-ish genes. So also the function of genes are much better annotated. You can try to validate in other model systems. Reverse causality that I mentioned before is not such a big of an issue. And also the direction of the effect. If you know that a higher expression level of a gene is associated with high risk of a disease, maybe lowering that expression, downregulating the gene may be a good therapeutic strategy. So that means that the candidate genes can be good target for drug development. So for example, if one gene, is a causal gene and you want to downregulate, and that's good for you expect that to be beneficial for the patient, then if you do it for multiple genes, maybe it will be even kind of more effective, right? That's what this paper has done. This is not my paper. It was done by other people that used the software that we published before our paper was even published. So that was all good. This is just kind of similar idea. We looked at, this is preliminary data that we never published, but I find it useful to show. So if you look at what I'm showing you here is the PredictScan, so gene level association results between gene expression levels and risk of type 2 diabetes. And in black is what we should see for all the genes that were tested. And in blue, I'm showing you the results, the QQ plot. So on the y-axis is the significance of the association, so the higher the better. So in blue, I'm showing you the distribution of the p-values for the genes that were targets of drugs that were successful. So I claim here that, in principle, PredictScan results allows you to predict a success of drugs. So this has been helpful. And obviously, you don't have to stop with gene expression levels. You can predict protein levels, metabolites, isoforms, and people have been doing that, we and other people in the field. So of course, this method has many limitations. It's not very good at predicting genes whose expressions are not very heritable. And those are, as someone mentioned today, those are more likely to be more relevant for the disease, right? If the gene is deleterious, the selection is going to constrain this gene more. You will find fewer EQTLs, and then those are going to be more relevant, but those are more difficult to predict. Also, we know that predictions don't transfer well across ancestries. We have problems with rare variation because this is population-based. You need many people, but if it's rare, you don't see it, you cannot really predict it. So that means we need very large sample sizes, and the steady state of bulk EQTLs or expression levels may not be disease relevant, right? That's the general consensus that is building. So we asked the question, can we leverage all these advances in deep learning method to build our PredictScan 2.0, like the new generation? So what we did is we took Informer, we tried a few, like one more, but we ended up settling on this one. And Informer, as how many of you know what it is? Yeah, most people. So it takes DNA sequences. It's a deep learning model that has, 250 million parameters. So as a statistician, for me, that was a bit of a shock when I saw it. But anyways, I see it working, right? And the output is this matrix that is 896 means that covers about 120,000 base pairs of the genome. And it predicts, it has like 5,000, over 5,000 columns or tracks that it's predicting. So it covering, so these are data from ENCODE, round-up epigenome, and so on. So it has DNA CISC data or ataxic, very few ataxic, so 600 of them, histone modifications or source of histones, transcription factor, ChIP-seq experiments, more than 2,000, and cage experiments that give you gene expression levels. So how well does this perform? And this is from there, the paper from Avsek et al. And you can see that this is one window, that the one output. And you can see that these are 4 columns in that matrix that I showed you before. And in green is the observed value. And in blue, you can see the predicted value. So it's pretty impressive, right? These were obviously cherry-picked, but we looked at other regions and they look maybe not as nicely, but they look pretty impressive in my opinion. So what is Informer doing? Informer is trained to perform well across the genome, right? So it's trying to minimize across the genome the predicted epigenetic correlation or the mean square error of the predicted epigenetic track and the observed epigenetic tracks. So it's working in the genomic dimension, which is not really what I am interested in. I want to know whether a person that has a disease in green, let's say, versus a person, a control, have different expression levels or they have different, chromatin accessibility and so on. So I am interested in the orthogonal dimension, right? So a method that has been trained to maximize correlation across the genome, there's really no reason, like statistically speaking, that it will do well to predict variation across individuals. So I didn't expect this. First time a few years ago, when I looked at these methods, I thought that it would seem promising. They seem to be the same thing we were doing, but I realized that this different dimension that we are looking at. So more recently, when Informer came out, we tried again. And then, so these are my students who tried this. They took the some 1,000 genomes where we have gene expression, observed gene expression data. They plugged in the DNA sequence, the personal DNA sequence of these individuals into Informer, predicted expression levels in LCL, so lymphoblastoid cell lines. And you can see here, y-axis is Informer prediction, and x-axis is the observed RNA-seq data from the GWAS data project. So the dots here are people, right? We are not looking at variation across the genome. We are looking at a given gene. So this is a specific gene. I forgot to put the name there. Variation across individuals. So this is, I was very impressed with this actually. And then, some genes we do very well, and some genes, we see this. So any problem with this? Yeah, okay. So, it's still significant correlation, right? So there was a flurry of paper coming out in Nature Genetics, and I found out that actually the Informer paper was rejected at Nature Genetics, and then they published these papers kind of criticizing Informer, which I thought that, so if you use Informer, I know not to submit it to Nature Genetics. Anyways, okay, so, Peter Koo, who was, I don't know, he's here, he was here yesterday. So Sarah Mustafabi and Naila and their groups kind of showed that exactly this problem, right? You see negative correlation as well as positive correlation. So anyways, if you, the issue I find with these papers is that, in general, people, myself including, we read the headlines, right? So the headline is these methods don't work across individuals, so you dismiss them, right? So, but if you look at the paper, this is in one of the papers, the Naila's paper, they also talk about the correlation across genes and across individuals. So across genes, the mean correlation is pretty good, and former that's better than all these other methods. But if you look at across individuals, the prediction performance is close to zero. But when you take averages, it's usually it can be misleading, right? Because you are looking at things that are positively correlated and negatively correlated, but significantly negatively correlated. So if you average them, you're going to see to have a zero performance, but that's not telling the full story. And this figure here is showing, so here the dots are genes, right? So how well a gene is predicting in terms of Pearson correlation. So there are positive performance and negative performance. So the x-axis here is the PredictScan performance. So you can see PredictScan is doing very well compared to this, but it's really not a fair comparison because PredictScan is trained to maximize this correlation, whereas these other methods are trained to maximize something completely different. So, but it was good press for us to have PredictScan to be better than other. Anyway, so, but the way I look at it is this glass half full or half empty? And you can look at the glass of water. I found this XKCD. I mean, oh, water, we can drink it, or drinking fluid, only postpones your death, right? So, but, we decided to drink the water, right? So I'm going to show you a few examples where, depending on how we're doing with the time, where we are using Informer to make it useful for what we have been doing, which is to try to understand what is the function of GWAS loci. So TF scan. So what are we doing here? We're trying to predict transcription factor binding. So why would one care about predicting transcription factor binding from genotype or from DNA sequence? So the general consensus, as I say, is that GWAS loci affect their phenotype through maybe altering transcription factor binding that will alter downstream gene expression. So this is from this review paper from Frank Albert and others, where they show in this example, for example, for this person that has the blue allele here, allows this transcription factor to bind very easily. That leads to very high expression levels of this gene. That downstream leads to high protein levels, and if the gene is deleterious, that will cause high disease risk, whereas this other person has the yellow allele, doesn't allow the transcription factor to bind as easily, the expression levels are lower, protein levels lower, so that leads to low risk of disease. So then why not predict transcription factor binding at the GWAS loci and see which one is the causal one, right? So there are tons of them. So that's what we try to do. So we'll try to, like a TWAS-like approach, where instead of predicting gene expression levels, predict transcription factor binding. So that's what I wrote here, predict transcription factor binding from the genotype, and then you can correlate with the phenotype. That will give you, for each transcription factor binding site, it will tell you whether this transcription factor at a given tissue is associated with the trait, with the complex trait. So, okay, how many of you know what is today's typical GWAS sample size? Is it like 1,000 or 1,000,000? Hundreds of thousands in between. Okay, so the largest one is several millions, right? Like for height, that was recently. So if we want to do this, right? Sorry. I'm missing a slide. So if we want to do this, okay. Yeah, let me get back to that later. No, yeah, sorry about that. So before getting into that detail, so that's what you can do, right? What we want to do is predict the transcription factor binding, and with the transcription factor binding, we can find those loci and identify the transcription factor that's culprit for the GWAS locus. So that's the goal. And what has been the field doing so far, and it's kind of, there's this method that was published in 2022 called GWAS, system-wide association studies. And they do exactly that, but they do it for androgen receptor in prostate tumor. And why do they do it only for that transcription factor tissue pair? Any guesses? Because this experiment, they need 130 samples, or about 100 samples, at least 100 samples, right? These are ChIP-seq experiments. So ChIP-seq experiments, 100 of them is hundreds of thousands of dollars. So if we want to do this for thousands of transcription factor tissue pairs, it just doesn't work, right? So they did it for this, which was good because now we can use this as our validation thing. So what they did, again, so they assayed 130 prostate tumors, and then they, with ChIP-seq experiments, and that will give us this matrix of transcription factor binding sites as columns and people as rows, right? So they use the same assays to call the genotype data. So they also generated genotype data from the same experiment, and then they trained this linear model on genotype to predict androgen receptor binding. So yeah, as I said, running ChIP-seq experiments for many samples is very expensive, so that's why they did it only for this and not many more. But what we would like to do is run this kind of associations for thousands of transcription factor tissue pairs. So that's where we propose this method that we're calling NPACT. And before explaining what NPACT is, let me explain what IMPACT is. So IMPACT is a method that was published in 2020 and some previous paper as well, where Tiffany and Mariuta, our collaborator, used epigenetic features to predict transcription factor binding using just logistic regression type of approach, some regularized because we are talking about thousands of features. So she did that very successfully and so on. So what we decided to do is we have Informer that will give us epigenome. We have IMPACT that from the epigenome will give us transcription factor binding. So we put them together, we call them NPACT. We're very creative with this. And then the ways that you need to learn here are learned from public data. So we use SystemDB. So I'm not going to go through the details. So if you look at the performance across the genome for like FOXA1, androgen receptor, GATA2, and HOXB13, all in prostate, you can see that the prediction is doing pretty well, right? So on the left, this thing here corresponds to unbound, and this right side, the darker green corresponds to bound, right? So we are predicting pretty well the difference between the NPACT predictors give you much higher values for sites that were bound compared to sites that were not bound. So that's very good. But again, this is across the genome, which is not really what we are interested in. So what about performance across individuals? So we just take different people's genotype or their DNA sequences, run through Informer, use the NPACT weights that we train with previous data, and we calculate NPACT prediction and compare to observed. So that's what you are seeing for one site. It's obviously cherry picked. You can see our prediction, and here the dots are people. So left side is people whose ChIP-seq experiment did not show binding of androgen receptor, and on the right, people who did show androgen receptor binding. So for this one site, we do pretty well. How about across the genome? So we did this for 18,000 different sites that our collaborators, Silvan Baccaral, had generated androgen receptor prediction based on population level data. So here, the way to read this is that X-axis is expected if there was no relationship between our prediction and the actual binding, and y-axis is the observed p-values, and you can see it departs from the null, so many of them is showing that we are significantly predicting the transcription factor binding. It's not as nice as I would like, but if we compare to how GWAS, right, is doing, it's our current state of the art, we do better than GWAS, which used the 130 individuals. We only use public data that were out there available. So that's that. So this is, the thing is, transcription factor binding is dynamic, so the observed value has a lot of noise, right? So what we are interested in is not actually predicting the observed value, we are interested in predicting the genetic component of it. So can we test that? And one way to kind of test that is to take GWAS, which is an approach that uses population dimension approach, and then our approach uses genomic dimension. So these are orthogonal predictors of the same idea, the same thing, androgen receptor binding for these individuals. So we compared the two things, and the way we did it, we took, again, 1,000 genomes genotype, completely publicly available. We applied it MPAC, we got this, rows are people, columns are androgen receptor binding prediction with MPAC method, and on the right side, we have at the same site for the same people the GWAS method, which is the current best of the class. So here I'm on the right, I'm showing you the comparison between our prediction, MPAC prediction, and the GWAS prediction, and you can see it's pretty strikingly similar, but again, I'm showing you only one site, right? So about the other sites. So on the left side, I'm showing you the p-values, because that's more, that's better, I think. It looks better, but it's the same data. So the p-values, most of the 18,000 sites that we tested, we get p-values that are way below 0.05, right? So clearly we're predicting, they're very, very similar, right? There's high concordance between our prediction with NPAC and GWAS prediction. And on the right side, showing you under the hood, the correlation between NPAC predictions and GWAS prediction. So the distribution is, again, you have negative correlation, positive correlation, but if they were not related at all, you would see them concentrated around 0. But instead of that, it's spread across. We have kind of some bias towards the positive side, probably because transcription factor binding prediction is easier than expression prediction. That's my guess. But in general, so what we're going to do is ignore this sign issue, right? So I used to work in finance. And in finance, if you have a strategy that systematically loses money, you can turn it around and make money, right? So this is the same idea. We're doing the same thing. If this thing is significantly negatively correlated, I just multiply by -1, and it will be significantly positively correlated. I don't know it's going to fly with reviewers, and but I'll show you more results to show that are very promising. But and the other thing is in TWAS or PredictScan or TFScan, as we're calling this, if you're interested in the p-values don't care about the direction of the effect, right? So we would also like to have the direction, but just having the p-values is pretty important. And the other thing is, in order to generate this thing with GWAS, they had to spend $500,000. We did this with a few $1,000s of compute time. So it's kind of, there's a huge difference, right? And this enables us to do things that I'm going to show you soon. So next, this is what I got kind of hung up before. So now that we have a way, we have the genotype data, we run it through our MPAC prediction, and we'll have predicted transcription factor binding. So, and then we can associate with the phenotype, and that gives you association results. So as the question about how big a GWAS is, right? Typically hundreds of thousands of samples. So running one run of the 4 pass of Informer is, I calculated roughly 1 GPU second. So if you want to do that for 1 million people, that's 277 GPU hours, which you could do it, but it's for one site, right? If you do want to do it for more sites, then it's just not feasible, right? So with current GWAS sample sizes, you cannot use individual level data. So if you come up with, and I see that all the time, especially among statisticians, they give you this beautiful Bayesian method that uses individual level data. I mean, it's not going to be practical because, we know the GWAS effect sizes are tiny, so you need very large sample sizes to discover anything. So any method, if you decide to develop methods that will explain GWAS loci, it will have to use summary statistics. So in order to use, so the idea is that it's not very hard to figure out that given the GWAS results, so these are what we call summary statistics, you can infer the result of the association if the prediction models are linear. So we need, we say, okay, we're using deep learning, but deep learning is clearly not linear, right? So okay, so that's what we need. So in order to make it useful, we need to make this model linear on SNPs. So how do we train linear predictors? Any ideas? Sorry? Well, interpretability would be nice, but just, I need to predict using a linear combination of SNPs. That's what I need. But the NPAC method that I have is not linear. It's this complex, informer plus. Anyways, so okay, I'll give you the answer. So what we do is we take, again, genotype data, 1,000 genomes, for example, and then predict the transcription factor binding using our MPAC method. So we generate in silico reference data. So once we have the in silico data for a number of individuals, we can just use the methods we've been using over the last 10 years, right? Because now it's back to what we used to do before. We have population level data, but this population level data was created in silico based on the deep learning model. So that's a trick we need to use if we want to be able to apply this to GWAS data. So that's what we did. And how well does the linear prediction compare to the full nonlinear predictors? And here are a few examples, and you can see it does pretty well, right? And on the right-hand side is the correlation across all 18,000 sites that we had tested. So, the correlation here is pretty close to 1. So, okay. So which also kind of reinforces the idea that transcription factor binding prediction is not that hard, it seems. Anyways, now that we have linear predictors, we can apply the PredictScan, summary PredictScan, all the framework that we had developed over the last 10 years to this data, and we can come up with this Manhattan plot-like results where x-axis is like, you have the different chromosomal positions, and each dot is, in this case, androgen receptor binding in prostate for all these at all these different locations. So, large effects are good and so on. So is this real, right? So then we can compare our results to GWAS, which was based on, approved population level data. So we can see that the z-scores are pretty similar, and we have to take the absolute value because we know that the direction is wrong. And so out of the 74 GWAS loci that were found to be significantly associated with prostate cancer risk, we find 40 of them are also significantly, Wolfrani corrected, like this is very stringent, significantly associated with our approach as well. So I would claim that we have a pretty good concordance, and I think even with this black box and negative signs and all that, we are able to detect most of the signal that GWAS finds. Okay, so next, this is completely new data. Sofia Salazar sent me these results this morning. So we applied this method. So Sofia generated prediction models for many more transcription factors in a few tissues, and she applied them to ovarian risk GWAS. So this is what you get. we're calling this Tenerife plot. So, the same way in a GWAS, you have Manhattan plot here, because we have many different transcription factors and then different loci. So it's two-dimensional instead of one dimension. We are calling this Tenerife. It was Temi, the graduate student who built this pipeline, called it Tenerife. And this is the world premiere. This is the first time we are presenting this figure. So, hopefully people are going to use it down the road, and you can say, I was the first one to see it. Okay, so how to read this? So on the x-axis are the different tissues, transcription factors, and in different tissues. So breast, cervix, because we are interested in breast cancer and ovarian cancer, these were the tissues that were used. And then in red is the GWAS p-values. So the more red, the more significant it is. And in blue are the p-value of the association between predicted transcription factor binding and ovarian cancer risk. So this was, and in the double star means that they are associated with a z-score greater than 10. So that very highly significant associations. So, these are, we just got these results. We need to kind of try to understand what that means. So our naive idea was that we would build this model, run this, and we would have one transcription factor, D transcription factor in the D tissue to be significant, and everything else is not, right? So we want it, but obviously life is more complicated. But still, I think this is very interesting results. It could be telling us that there's a lot of cooperative binding. It's not. We also found that these associations are not mostly driven by motif alteration. So there's a lot to be investigated, but we are very happy with what we have so far. And this is the first time that anybody, as far as I know, right? The only way you can do this is using this pipeline that we use using the in silico and former predictions. There's no way you can generate all these population level transcription factor ChIP-seq experiments in hundreds of people. This is just, try to see, do we see, the other thing is when we kind of thought about creating this Tenerife plot, we were not sure, would we find some transcription factors that would be sort of always significant across all the loci, GWAS loci, or what we're finding is more like, horizontal patterns and not vertical patterns. But then maybe some transcription factors come up over and over. And this is what we are finding. We need to consult with our collaborators who know a lot more about ovarian cancer biology. And also, I would really appreciate your input about what you think about these results. CTCF, I don't know whether it's a good news or not good news, but anyways, that's what we have. So in summary, the TF-scan approach, transcription factor binding prediction, is an extension of TWAS that uses predicted transcription factor binding. We use NPACT, which is Informer plus Impact, to train transcription factor binding in tissues in a tissue-specific manner using just public data. These NPACT predictors can be turned into linear functions, and the linear functions of SNPs, they work pretty well. And then comparison with the state-of-the-art CWAS is validating our predictions and the association results that we are finding. So now applied to ovarian cancer using many transcription factor tissue pairs, nominated transcription factors involved in ovarian cancer pathogenesis. How am I doing with time? I used up the time. Okay, so I'm going to skip the single cell application. And the take-home message, the take-home message. I just want to. I don't want to. You want to leave. So no. So I think we came up. We used Informer, which was heavily criticized, but I think it's kind of unfairly criticized. You can use it to kind of infer population level variation data, and the fact that we can do this, it means that you can train across the genome, right? You only need one sample to do that. So that means that going forward, when we design EQTL experiments or single cell EQTL experiments, we are not going to need that many samples, so that this should inform like the design of future experiments and we should try to cover more, context, more ancestries, more perturbations. And even, if you have extra money, use more reads instead of more samples, right? We do need a few more samples. I'm not saying we can get away with just one sample, but, given budget limitations, it's better to go broad than, have more samples, more individuals. So thank you very much. Yeah.
Thanks for the wonderful talk. That was great. Maybe I missed this, but the NPACT model, am I correct in saying that you're using Informer to predict the histone modifications, and then you're using IMPACT to predict TF binding?
Yes.
So why not use Informer directly to predict the TF binding?
Because Informer has many tracks, but it doesn't have, for example, androgen receptor binding in prostate tissue. And also, even if it was there, using more information gives you better prediction, actually.
Okay, but you could, in principle, train another transformer to...
You could.
On
yes,
special
yes.
chip-seq data that you would... Okay.
Yes.
And then one other thing, and when you get lots of predictions for one locus, just like you were showing in the Tenerife plot, it's possible that maybe that SNP is like creating something that's more of a promoter, and now all of these other TFs are associated with promoter activity as opposed to that they're all affected by the SNP. You know what I mean? That this SNP that you're introducing might... It's
perfectly possible. We don't know enough about transcription factor biology to figure that out, but I would love to talk with you about that.
Okay, thanks.
So fascinating. I love the Tenerife plot. So how are the columns sorted here? I might have missed that.
It's heat map.
Oh yeah, but is there an okay?
Columns,
so it's
these
hierarchical
columns.
clustering.
Yeah,
okay.
Yeah.
Huh. Okay. Thanks.
I mean, yeah, it's yeah.
I have two questions.
Yes.
So the first one is
one at a time, please.
One at a time. Okay, good. So this is very simple. The first one,
Yeah.
Informer gets like a linear sequence, and you and you have variation data. So it might be heterozygous or homozygous. So how do you use Informer with your data?
So Informer takes in any sequence you give it.
Yeah.
Right.
So in one hot encoding, then sometimes you have a one at A and C, and sometimes only at A.
Like so the way Informer was trained, they use reference genome, right? But now nobody is the reference genome. So if you want to calculate the personalized epigenome, you plug in the person's actual genome and run Informer, and it'll give you this.
But you have two alleles.
You do both, and you average them.
Okay. You do it twice. Okay,
Yes.
I see. Now, if for you basically said, and Informer takes so long, you can't use it in a way you really want to use it, right? That's how I understood it.
So it takes 1 GPU second per matrix. It's a lot. It depends on what you want to achieve. We have been running, many of these. You cannot run a GWAS type of thing for 1 million people, but you can run. We have this calculated for all of the 1,000 genomes individuals, for example.
Yeah. No, I mean, I agree. It's too expensive right now. Maybe in 5 years when computers are even faster. And so now, just that I understand. So now you take for one locus and one TF, you build a new linear model that just predicts how does the genomic variation change the binding, no binding at this site. So you have like TF times number of sites, many linear models that are actually part of this TFX scan.
Yeah, so that's a problem, right? So with GWAS, you have genes. You only have 20,000 TSS, and that's it. With transcription factor binding, where do you focus, right? That's an excellent question. What we do is we think of one GWAS, right? Ovarian cancer, it has 30 loci. I don't know. With breast cancer, maybe you have a couple hundred. So we actually find map. So what's the most likely causal SNP? and then we run Informer around that causal SNP, because our goal is to explain the GWAS locus, the GWAS hit, right? So we center at the GWAS hit. So this is specific. You have to train this model for each one. That is a little bit costly, but still kind of, not comparable to $500,000.
Yeah. No, it's really beautiful. I just wanted to understand the rationale.
Yeah, no, there's a lot of detail. It's not that it's hard, but kind of there are many details that when kind of, if I tell you all the details, you'll get bored, right? So,
So let's think how